Good morning, everyone, and thank you for joining us this morning for today's installment of the Cape Perpetua Speaker Series. Today, we will be hearing about Oregon shellfish in an acidified ocean with Stephen Rumrill. And our next presentation is next Saturday, January 15th. We will be hearing about ecological aspects of transient killer whales off of the California and Oregon coast with Josh McGinnis. I'd like to acknowledge that the Cape Perpetua area landscape from Yachats to Florence is the traditional territory of the Siletz tribe and Coos, Lower Umpqua, and Siuslaw tribe. And we want to acknowledge that the tribal governments and their roles historically and today in taking care of these lands. You can learn more about each of these tribes on their respective websites. A little bit about the Cape Perpetua Collaborative. My name is Tara DuBois. I'm the communications coordinator for the collaborative. And our vision is to foster conservation and collaboration within local communities for scientific exchange, management, awareness, and stewardship from the land and sea in and around the Cape Perpetua Marine Reserve. And our three guiding principles are community engagement, leveraging resources, and engaging in partnerships. And you can see along the bottom here a variety of logos. These are our founding partners who pulled together the collaborative in 2017. But I also like to acknowledge that we have a variety of other local partners, cities, and businesses that we also partner with, and that we cannot do this work without any of our partners. We appreciate working with them. The Cape Perpetua Marine Reserve and protected areas stretch from Yachats to Florence. Cape Perpetua Marine Reserve is Oregon's biggest marine reserve. So there's some form of protected waters that stretch between Yachats and Florence when you include the north and southern marine protected areas. Oregon Department of Fish and Wildlife is the agency that oversees the monitoring of the marine reserves in Oregon, and there are 5 total. We also host a variety of community science within and adjacent to the Cape Perpetua Marine Reserve. You can see here the list of projects. Many of these are seasonal and partake like spring through the fall. We do host monthly beach cleanups. And in addition to this speaker series, we also host a Young Scientist webinar series. That's the second Tuesday of the month, October through April. And you can engage in BioBlitz at any time, which helps document biodiversity using the iNaturalist.org app. So if you have that app downloaded on your phone, or if you don't, I encourage you to learn more about it and download it. Do connect with the Cape Perpetua BioBlitz series, and any observations that you make that are within that footprint will automatically upload to our project. And you can find out more information about all of our events and our presentations that we have coming up on our website at capeperpetuacollaborative.org. And I always like to encourage folks to connect with us on our Facebook and our YouTube page as well. If you like the work we're doing and you feel inclined to donate, on our website, there's also a donate button. And if you can just click on that, and that will take you through the steps. Today, I'd like to highlight that it is the 4th International Ocean Acidification Day of Action. So January 8th, or 8.1, which reminds us of the current pH level of the ocean. And the Ocean Foundation, we're partnering with, in addition to the Oregon Coast Aquarium and other agencies and nonprofits in the state, to highlight ocean acidification today. And the foundation is committed to finding solutions to the crisis. And you can read more about this initiative on the Ocean Foundation's website. And I've listed that here for you. And I can also put that in the chat as well, so it's easy to click on if you'd like to check it out. And with that, I'd like to introduce our speaker today, Stephen Rumrill, Shellfish Program Leader. Oregon Department of Fish and Wildlife has statewide responsibilities for management of multifaceted program that encompasses policy development, resource assessment surveys, commercial and recreational fisheries, scientific research, and outreach activities for a diverse group of shellfish that inhabit Oregon's bays and estuaries, sandy beaches, rocky shores, subtidal reefs, and deep water habitats. Before joining the ODFW Marine Resources Program, Steve served as the chief scientist and research coordinator for the South Slough National Estuarine Research Reserve in Charleston, Oregon. He holds adjunct academic appointments at the University of Oregon and Oregon State University, where he conducts scientific research in collaboration with faculty members and serves as an advisor to graduate students. Steve received his master's degree in marine science from University of California at Santa Cruz in 1984 and his PhD in zoology from the University of Alberta in '87. And with that, Steve, I'm going to stop share and you can bring your screen up. And while Steve brings his screen up, I just want to let you know that you are welcome to put in your questions into the chat box or the Q&A box throughout the presentation as they come up. And after the presentation, we will hold a Q&A session and address the questions. And
all right, thank you so much, Tara. And I want to just say thank you to all of the members of the audience here for taking time out of your busy schedules and listening to what we have to say today. It's truly an honor to be selected as speaker here on this 4th International Ocean Acidification Day of Action. And so I'll embrace the memory of this talk, trying to hang on to 8.1, trying to do what we can to maintain healthy oceans and to retain a normal pH of 8.1. So I'm going to speak with you today about ocean acidification and Oregon shellfish. And the story that I'll tell will really just describe an overview of the emerging threat posed by the dual stressors, ocean acidification and hypoxia. So as I get started, I'm going to talk about the big picture and really just paint this picture with a broad brush and not get too far down into the seaweeds. So once again, I'm Steve Rumrill. I'm the Shellfish Program Leader for Oregon Department of Fish and Wildlife. My co-author today is Dr. Charlotte Regula Whitefield. Charlotte is our Ocean Acidification and Hypoxia Policy Specialist, and she really is the go-to gal for tracking the activities within the state of Oregon and throughout the region to address this emerging threat. So let's get started. I'm gonna move at a pretty good clip here. Let's see how this works. Well, Tara, I'm having trouble advancing.
Sometimes I have to click on my initial screen before it will advance for me.
See if we can get through the tech problems. There we go. Okay, cool. So here's the outline. I'll speak with you first about the fundamentals about upwelling and ocean acidification and hypoxia. Spend the bulk of my presentation here today just describing kind of case histories of what's known about the impacts of ocean acidification on shellfish in Oregon or Oregon shellfish at risk. And I'll finish off with a couple of examples of possible biobuffering of acidified marine waters, promising thing there. And I'll also wrap up just talking a little bit about policy actions to address ocean acidification and hypoxia, and describe really quickly the pathway for a sequence of Oregon initiatives, very proactive work here in Oregon, describe the new work undertaken by the Oregon Ocean Acidification and Hypoxia Coordination Council, and then just finish with a quick description of recent actions to address OAH here in Oregon. And then we'll wrap up with opportunity for questions. So let's get focused here. So Oregon's marine and coastal ecosystem that we're all familiar with is really that biogeographic transition zone between the subarctic, Colombian, and Californian bioregions. We have a very rich and productive section of the coastline that we call home. Our area of the coastline is characterized by seasonal upwelling that drives high productivity rates. We have summer winds and fog, that strip of fog that keeps our coastline cool. And upwelling intensity, of course, is not the same all along the coast. It's heterogeneous along the coast. Some areas have high upwelling and other areas not so much. This zone along the coastline is really characterized by high biodiversity, major groups of marine organisms, including marine mammals, high diversity and productivity, typically of seabirds and shorebirds, of pelagic fishes, ground fishes, and then my realm, a high biodiversity and productivity of marine invertebrates. So most of us are familiar with upwelling, how that works. I'll just describe that really quick. So wind-driven upwelling occurs along the Oregon coast. The winds deriving typically in the spring and summer from the north to the south create wind stress along the coast. Oops. And due to Earth's rotation, that the water is displaced offshore, and that leads to upwelling of deep, cold, nutrient-rich, low dissolved oxygen, low pH waters well up from the deep up along our coastline. Last point I want to make here is that these upwelled ocean waters are undersaturated with respect to calcium carbonate. We call them low omega or low saturation waters. And these can be dangerous to organisms, marine organisms that incorporate calcium carbonate into their bodies. So the upwelling brings us cold, low dissolved oxygen, high nutrients, low pH, and low calcite, aragonite saturation state waters up to the coast. The image on the right, the map on the right, just shows the image of the coastline from Vancouver Island down into Baja, Mexico, just to illustrate that we are really a focus for cold waters. Those blue cold waters that characterize the area off Vancouver Island also upwell along the coastline of Oregon, Northern California, and down into Central California. Coupled with ocean acidification, of course, is the problem of hypoxia. And hypoxia is low dissolved oxygen in waters. Over the past decade or so, we've come to the realization that we're seeing more and more hypoxic waters, greater frequency, longer duration, greater hypoxic events as well. This graph in front of you here just shows depth from the surface at 0 down to 800 meters along the left side, and then concentrations of oxygen, with low on the left, no oxygen, to low 1 to 2 parts and there up to high. So typical waters are high and have high concentrations up in that 5 to 6, maybe down to 4 up on the surface. But as you get deeper and deeper down into deeper water, the level and concentration of dissolved oxygen drops way down. And that was a situation from measurements from 1950s up to about 1999. In that period from 2000 to 2005, we began to have measures that were dipping down into what we call the hypoxic transition zone between 1 and 2. And so the green value showing up there. And then from 2006 on, really now having these episodes of very hypoxic waters up in the shallow surface area. It's alarming. And our colleague and friend Francis Chan published this work way back in 2008 when we were just making this discovery. Here's an example of another graph that just shows the hypoxic season. So the graph in front of us shows time. So there's March, April, May, June, July, August, September. And the typical situation with normal oxic waters in that area of around 4 milliliters per liter or 3 millimeters per liter. But the concentrations of oxygen in our nearshore waters dip down along the Cape Arago or Cape Perpetua coast and began to make that transition into hypoxia at 1.5, 1.4, about in typically late April or May, and then enter into that hypoxic season that can extend from May through August and then transition back up in September. Just wanted to point out here in red that the 2020 observations by our PISCO colleagues was a little bit delayed, but really dropped down into very hypoxic waters in mid-June and really reached the bottom in August and early September before finally coming back up. So we now have that situation where way back in 2007, discovery of this low dissolved oxygen zone, hypoxic zone, and now here in 2018, 2020, recognizing that we have really developed to the situation where we now experience a hypoxia season, just like a wildfire season. Pretty alarming. Well, what does that mean? So this triangle graph here just shows levels of dissolved oxygen, so 7 to 8 milligrams per liter, down to 3, 2, 1. The yellow shows that dangerous transition zone, where above that, in that 2 to 3, it impacts to mobile species, but mobile species are able to move around and readjust their location to avoid hypoxic waters. But the non-modal species that don't have that ability to retreat or relocate themselves, they begin to really suffer in that zone of about 1.5, 1.4 concentrations of dissolved oxygen. So below that, we have trouble, and that's when we get crab die-offs of sea stars and sea anemones and shrimp. And then below that, we're really down into to very low dissolved oxygen levels, which are, of course, very detrimental and can be fatal. All right, let's shift focus over to ocean acidification. So this graph shows the time series of increasing atmospheric CO2 values in the atmosphere. So the red line just goes way back to my birth date, 1958, up to 2018. And you can just see that steady rise in the concentration of carbon dioxide in the atmosphere. This is measured out in Hawaii, which should be pretty open and free from terrestrial pollutants. So you can see that the concentration of carbon dioxide in the atmosphere has just steadily increased over this 65-year period. And the measurement today, I just checked it this morning, so we're up at 418 parts per million at the beginning of 2022. So that rise just keeps on going up. So as atmospheric CO2 has gone up in the red line, so has the amount of CO2 dissolved in the ocean, the PCO2 of dissolved carbon dioxide in ocean waters. That's shown here in the green line. It's highly variable, but also shows that steady increase from the beginnings of measurements there at about 1988 through 2020. And then coupled with that, you can see in the blue line, the inverse relationship, the decreasing pH of ocean waters from 1988 down to about now. And again, here's that pH level on this side where healthy ocean waters are at about 8.1 or so. So we're making that transition from healthy ocean waters to not healthy ocean waters. That's alarming. And this decreased alkalinity or increased pH, decreasing pH is ocean acidification. So this graph shows that same kind of situation. So there are spatial variability in the pH of surface waters along the Pacific Northwest coast. The map here shows Vancouver Island and Strait of Juan de Fuca. The blue values show healthy ocean waters, 8.1, 8.2, 8.3. But you can see these unhealthy ocean waters shown in red and orange off the mouth of the Columbia River, reflecting the very strong influence of the Columbia River plume extending out into ocean waters. But then you can see that persistent orange haze extending down the coastline of Oregon off to Newport and here at Cape Perpetua. We do have unhealthy ocean waters in the surface occurring all along the Oregon coast here up from Cape Perpetua north. So, and also you can see as you extend that down, you begin to pick up that haze of unhealthy ocean waters at some of the major headlands, Cape Mendocino and Port St. George, and extending on down. So a concern up along the coast. So ocean acidification is a direct result of increased carbon dioxide emissions into the atmosphere. This cartoon just shows a cloud of atmospheric carbon dioxide in the atmosphere, diffusing down into the surface waters. So CO2 in ocean waters is going up, and that disrupts the carbonate chemistry of ocean waters. So bicarbonate values, as we go through these chemical reactions, bicarbonate increases and carbonate decreases. And this drives the hydrogen ions up and pH down. So the take-home message from this chemistry is that increased carbon dioxide in the atmosphere contributes to lower pH in the ocean waters. Another way to look at that is in this cartoon on the left, a healthy situation characterized by the late 1800s till now that experience really characterized by reduced acidity. You can see that on the pH scale about 8.1, 8.2. lots of carbonate ions and healthy, abundant organisms that incorporate calcium carbonate into their bodies, like corals or mollusks and other calcifiers. In contrast, here projected out roughly 80 years from now or so, the decrease in seawater pH below that healthy value of 8, down into 7.9, 7.8. And in that situation, fewer carbonate ions available, and the expected result are fewer, smaller marine organisms that rely on calcium carbonate. Another really potent graphic picture, if you just look at this from left to right, the Pleistocene or time frame of glaciers and woolly mammoths, pH was thought to be about pH 8.2 at that time. And then with the onset of the Industrial Revolution over the last 100 years, really dumping lots of CO2 into our atmosphere, that CO2 has changed the carbonate chemistry of seawater and has contributed to a decrease down to pH 8.1 from 8.2, and expected out into the future that will continue to drop 7.9, 7.8. And you can just see the graphic on the bottom of healthy marine communities now making that transition to unhealthy marine communities. Okay, so what we need is to take those kind of graphic images that we have developed for other parts of the world and think about what does that mean for our coastline here that we all know and cherish so well at Cape Arago. Cape Arago is a coastal land margin ecosystem influenced by winds, by upwelling, by the ocean waters and the global atmosphere, but we also have our own regional climate conditions and conditions. And in particular, when you move from thinking about the nearshore ocean waters to that nearshore transition zone, you can just see that broad band of highly turbid, highly productive waters here, looking down from Cape Arago, where the water becomes turbid, becomes agitated as the swells break along the shoreline and make that transition from an ocean ecosystem to a terrestrial ecosystem. So some very interesting kinds of dynamics and complicated transitions going on in this location. We're just learning about this. So I'm not going to go through chemistry again, but I just wanted to make that point again, that the elevated CO2 dissolved in seawater causes the seawater to become undersaturated with respect to carbonate and to become more corrosive to the shells of marine organisms. Now let's look at some of the detail of these marine organisms. So on the left here, just a photograph of a clam, just showing that they've got a dorsal and ventral side and an anterior, posterior. Just want to make the point that the clam shells are produced by layers and layers and layers of calcium carbonate being laid down by the mantle or the soft tissues inside. So the cartoon on the lower right just shows that layering on the outside at the top. The periostracum is that thin kind of layer right at the top, and then the hard parts of the shell, external, moving down into that nacreous layer, or that kind of translucent mother of pearl layer that we know so well in abalone and appears in mussels as well, down on the inside, and then the soft tissues responsible for secreting those shells. So there's layers and layers of calcium carbonate being laid down to create these shells. Mollusk shells are composed of calcium carbonate that occurs in two different forms. It can occur as calcite, which is less soluble in water, but also in the form of aragonite, which is more soluble in water. So these are known as polymorphs of calcium carbonate. And I won't go into the structure here, but if you look at the SEM photos on the right, you can just see those plates of calcium carbonate, calcite and aragonite being laid down by the mollusks to create their shell. It's really almost like scales of calcium carbonate rather than a full solid structure. And they of course stick together, but they're laid out in these plates. If we look at a cross section through a Saxodomus or a butter clam, like we have along our coast, not this species, but again, you can see the difference between the outer layer, middle layer, and inner layer. And in this case, the outer layer, shown in blue in the graph, is composed primarily of the harder calcite, But as you move into the inner layer, that's where the composition of the shell changes to the more soluble aragonite, and same thing on the inside. So one of our leading stars in looking at all this, Dr. George Waldbusser at Oregon State University, published a number of papers looking at the health index and health matrices associated with these undersaturated waters, and put together this nice graph. So this shows pH across the bottom, healthy situation on the right at 8.3, 8.4, making that transition from 8.1 down to 8.0 and 7.9, and comparing that to the aragonite saturation state of seawater. And we have healthy shellfish occurring up in that range of 8.1, pH range 8.1 to 8.4. But then as we move down into these lower pH zones below 8, and at that aragonite saturation state below 2, we began to pick up unhealthy shellfish. So we're hovering right on that edge of making that transition from healthy to unhealthy. Okay, so let's focus now on what does this all mean? What do we know about Oregon shellfish and their risk due to ocean acidification and hypoxia? Again, there's lots and lots of species, great diversity of shellfish along the Oregon coast. And I'm just going to focus on very few of them at the broad brush level, but I'll just walk through and talk about several different ones. First, one part of the story deals with these beautiful, delicate marine snails known as pteropods. So these are mollusks, snails that swim and live out in the plankton along the outer coast in ocean waters. So healthy pteropods have a very fragile, beautiful shell, and they extend their soft tissues out and swim up in the ocean waters. And when they're healthy, that shell has sculpture associated with it. But when they're unhealthy, that very thin, delicate shell begins to break down and show signs of stress. So pteropods are good indicators of ocean acidification. And way back, almost 10 years ago or so now, began to recognize that these delicate shells began to showed evidence of stress, of dissolution, and that they could be used as an indicator of ocean acidification exposure. Here's a graph that just shows some more of that pteropods and their shells are healthy at aragonite saturation states above 2.5 or 3. They become stressed at around 2 and show signs of shell weakening and real stress down there at 1. Surface waters on the left, the graph on the left shows surface waters with higher levels of aragonite, but really lower levels, stressful waters down deeper below 25 meters, just showing us an indication that these cold, low dissolved oxygen, low aragonite saturation state waters are down deeper typically. Okay, also in the Oregon story, ground zero for understanding the impacts of ocean acidification on shellfish, discoveries made up at the Whiskey Creek Shellfish Hatchery. So this is a commercial shellfish hatchery that raises larvae of oysters, and they were having major problems with cultivating their larvae, and lots of good sleuthing up there figured out that their larvae were succumbing to ocean acidification. Again, more work by Dr. George Waldbusser, photographs there showing healthy petavillager larvae. This is the larval stage of an oyster during favorable carbonate conditions on the left, and then unfavorable carbonate conditions, which are difficult and stressful for these larvae. And you can see that larval shell is shrunk and disfigured and not healthy. So some major problems there with trying to raise oyster larvae in acidified ocean waters. This figure cartoon on the left just shows the life history of an oyster. So there's adults on the left releasing their eggs and sperm into the water, which develop into embryos and early stage larvae, later stage villager larvae, up into what we call a petavillager larva that has a foot and the ability to swim, that settles out back down onto the bottom, settling on another shell, for example, and then over a period of about 1 to 3 years growing up into a mature adult. So the image on the right just shows one of these villager larvae. It's got a small microscopic clam shell, but also the velum that extends out, covered with ciliary hairs that allows it to swim up in the water column. So these are the stages that are of great concern, these planktonic developing small microscopic larvae. So the great advances have been made in the shellfish hatcheries to set up monitoring systems to monitor the calcium carbonate in the water within these hatcheries. So they've got automated systems now that keep track of the situation and can add buffers and time the pumping to deal with the problems, not only for raising larvae, but also for raising the food, the microscopic algae that serves as the food for raising these millions and millions of larvae. Well, what do we know about the native Olympia oysters? So the Oregon coast, California, Washington, British Columbia also have a native species of Olympia oysters, Ostrelurida. And these animals, unlike the Pacific oysters that I just showed you, the adults here retain, the females retain the eggs, and they're brooded by the female internally in that protective mantle for those early stages. So the embryo, the early veliger and late stage veliger larvae all are retained within the female, and then they're hatched out at the later stages, the petaveliger that can both swim and crawl on the bottom, ultimately to settle back down onto the bottom and then grow up. So what we've learned by looking at those, that there's carryover effects from smaller shell sizes to the post-larval stage associated with ocean acidification. But George and his group have also shown that ultimately we end up with normal shell sizes, normal growth rate, and calcification rates in our native Olympia oysters. So it could be that our native Olympia oysters are more resilient, better adapted to deal with ocean acidification and the shifts and variability in the carbonate chemistry of seawater. What do we know about mussels? So mussels occur attached to rocky shores all along our rocky intertidal, and several species, Mytilus californianus, the California mussel, Mytilus trocylus. These, of course, we know are mollusks, but they're attached to rocks by byssal threads, photograph showing those byssal threads that are secreted by the mussels. They form multiple attachment points to rocks in these high energy surf zones. That's how they hang on in the surf. And experiments done in California and elsewhere have shown us that those byssal threads that are grown under elevated PCO2 values or acidified ocean water conditions, they exhibit slow growth, less flexibility, and decreased strength. And so with continuing ocean acidification conditions, we may expect to see decreased strength and flexibility and resiliency of those byssal threads, which could contribute to the detachment of mussels and some loss of structure in the intertidal zone, rocky intertidal zone. Let's shift to another very important and iconic shellfish here for us, and think about ocean acidification impacts to Dungeness crab. Dungeness crab shown on the left, Metacarcinus magister, and the red rock crab, Cancer productus, shown on the right. This series of photographs just shows the various life stages of cancer crabs. So here's an adult red rock crab. They hang on to their embryos and hatch. Ultimately, the females will hang on to them underneath their abdomen and then hatch and release planktonic zoea larvae. So these are microscopic to mesoscopic zoea that are up in members of the zooplankton. They undergo several molts and then will molt into the late stage megalops. That looks like a little crab with an extended abdomen sticking out the back. And then typically in late spring, they will recruit up into the shoreline, up into the bays and estuaries, sometimes so dense it's difficult to walk without stepping on them. But lots of these megalops and recently metamorphosed Dungeness crab shown in the lower left photo, and then they'll grow up into juvenile crab in the bays and estuaries, but all along the shelf waters. So a typical life cycle. So what do we know about the impacts of acidified ocean waters? Zoea larvae have been cultured at low pH waters, 7.5 and 7.1, and those that were put into those experimental conditions exhibited retarded levels of early development, slow early development, and also reduced rates of larval survival. That's for the Zoea larvae. And subsequent work I'll just get to is describe the impacts here for later stage megalopy. So already we know that these planktonic stages then could be impacted by increasing the amount of time that they're up in the plankton. And during that time, I won't go into it, but they are very subject to predation by fish, by gelatinous zooplankton, and others. So this retarded and slowed development certainly can contribute to reduced larval survival and greater rates of larval mortality. More recent work, same kind of time frame, back in 2016, has shown that the megalops stages, these late stage crab larvae, are also feeling the effects and stressed by ocean acidification. So what that has shown us there, these megalops stages and Zoea stages, the carapace or the test or shell surrounding them, they have very fine CT or mechanoreceptors that are up on their shell that are connected into their nerve systems. Those are very important for figuring out how to swim and orient themselves, and for prey capture and detecting the shear of water across the larval body. And those mechanoreceptors are corroded and damaged by acidified waters. This series of photographs shows a megalops larva on the left and the normal situation in panels B and C, showing the pits where these mechanoreceptors come out of the carapace and on the larval legs. But then in panels D, E, and F, which is increasing severity of corrosion due to acidified ocean waters, to the point where down in G, there's structural damage to that larval test, all the way to the point of illustrating or showing the exposed calcite crystals embedded in the test. So greater and greater dissolution. And here again, just a couple more photographs that show here right in the middle, one of those mechanoreceptor CD coming out of the leg on one of the crab larvae. And then just the pits and holes that have outrooted these CD have just come loose and disappeared. So we can expect then that larval swimming and ability to evade being captured and to capture prey, all of those kind of behavioral things may be then impacted by greater acidified ocean waters. Okay, Michelle Nguyen, a recent graduate from Oregon State University, also worked with her advisors in ODFW to investigate the impacts of ocean acidification on pink shrimp. She just gave a talk recently, so I won't go into that into detail, but pink shrimp is a very important shellfish here for us along the Oregon coast. Shrimp cocktail, shrimp salads, it's a big commercial fishery for us. And like Dungeness crab, they also have a series of zoeal stages. They're larval zoea, so a photograph of a larval shrimp shown in the middle. And Michelle's work showed that zoea larvae cultured at low pH values, 7.6, 7.8 or so, all the way through healthy waters and up to 8.2. She was able to show that the low pH reduced the growth rate in the zoea larvae, but there was very high variability in her experiments and non-significant statistical significance there, but she did show a trend there. And she was also able to show that there was no clear impact of lower pH on the respiration of zoea. Michelle also worked on the combined effects of ocean acidification and warming, and encourage you to look back at her presentation for those complicated results. What about echinoderms? So Oregon has a very active, productive fishery, commercial fishery that focuses on commercial harvest of sea urchins along the Oregon coast. And work has shown there, looking at the impacts of acidified ocean waters on the planktonic larvae of sea urchins. No strong significant damage to their larval skeleton, Sea urchins have a calcite skeleton. You can see the rods of these crystals inside the larval body. But they did document decreased size and growth of the larval urchins, and also the compromised expression of genes during larval development. They just didn't develop properly, and also disruption of larval digestion and their metabolism. So some impacts there. What about red abalone? Red abalone are very important species for us, an iconic species in the rocky subtidal zone down along the southern Oregon coast. So these are large grazing marine snails that occur in Oregon from Cape Arago down to the California border, and sometimes occur in the lowest intertidal zone, but they typically consume drift algae and seaweed and kelp. And the current situation, you probably know, is very poor for red abalone. So poor environmental conditions associated with the marine heat wave have contributed to very low densities of abalone, and we're seeing very poor reproductive success. So in response to that, the Oregon Department of Fish and Wildlife initially suspended the recreational fishery from March 2018 to March 2021, and we just recently extended that through March of 2024. What do we know about ocean acidification impacts to red abalone? So the findings for research carried out down at UC Davis have shown that red abalone that originate from areas that experience strong upwelling and low pH typically grow very slowly and are much more tolerant to ocean acidification. The photograph at the top shows abalone on the tip of a finger, and the very small one is the exact same age as the very large one grown in an abalone farm. These fast-growing farmed abalone are highly susceptible to ocean acidification. And it's possible, really, that this enhanced tolerance looks like it's due to a higher level of energy put into each egg by the mother, provided to the offspring in their yolk. And that is not afforded to the offspring raised in the farm situation. So very interesting and complicated result there coming up. Right now, we do have very poor environmental conditions for abalone along the southern Oregon coast. Mentioned that our kelp beds have been reduced at many locations, not everywhere, after the marine heat wave. Photograph just showing lots of kelp back in 2014 at Orford Reef off of Port Orford, and very little kelp in comparison to that same location in 2016. But this is complicated by other factors, very large numbers of purple urchins that are grazing seaweeds, and also sea star wasting syndrome. But photographs below just showing healthy abalone with lots of tissue, and a starved abalone with a very much reduced withered tissue mass. So overall, the big picture is that the impacts of ocean acidification on marine organisms are generally negative. So this graph shows the mean effect size from a big meta-analysis for various aspects of marine organisms. 0 shows that neutral zone, and above 0 is an increase in the function. Below 0 is a decrease in that function. So the way to look at this quickly is that ocean acidification causes a decrease in survival, a decrease in calcification, general decrease in growth, neutral on photosynthesis, a decrease in rates of development, an overall decrease in abundance, and a possible positive, looks like a positive, on the rates of metabolism. Overall, not a good picture. So there's lots of work ahead. We'd like to know the impacts of ocean acidification on lots of other shellfish. We don't have work to assess the impacts on razor clams or on soft shell clams, cockles, little neck clams, gaper clams, or butter clams here along the Oregon coast. And I'm looking forward to working with agency investigators and academic colleagues and graduate students, citizen scientists, to put together those stories so that we gain a better understanding of the impacts to these very heavily targeted species of clams. One other thing I want to just raise up, that the combined influence of ocean acidification and climate change and warming may have a positive impact on harmful algal blooms. I won't go into the details, but we saw a possible increase or neutral on photosynthesis. Typically, you think of winners and losers. The losers appear to be shellfish, but some of the winners may be some of these harmful algal blooms, which could have some tough impacts for us on lots of different shellfish. I won't go into that right now. I want to just start to wrap up by thinking about possible ways to deal with this. Eelgrass in the bays and estuaries is an important ecological engineer. We recognize that for lots of different functions, for being a primary producer of plants and source of detritus that's important at the base of the food web. Eelgrass is recognized as being important as a sediment trap and for nutrient exchange. Down in the water, eelgrass is recognized to contribute to water quality improvements and also to provide important habitat for juvenile fish and shellfish. Way back when I showed you that initial increase in carbon dioxide in the atmosphere, I didn't point out, but that steady increase over my lifetime also shows a seasonal pattern of high, low, high, low, high, low. Down here in the middle of that annual cycle of high, low, is really reflecting the global pattern of respiration and photosynthesis in the global ecosystem. If we look at that at the micro level inside an eelgrass bed in an estuary, just some of my early work way back 2008, you can see that same high, low, day-night pattern that reflects changes in pH, that reflects the photosynthesis and respiration by eelgrass and also macrobenthic algae. So during daylight, these plants produce oxygen and utilize CO2, and our values go up. And so that pH cycle is driven by photosynthesis and respiration by a complex group of phytoplankton, diatoms, macroalgae, and eelgrass beds. And a photograph here that just shows an example when we were working that out. Some of these big, dense, beautiful eelgrass beds can contribute to lowering CO2 and increasing pH. It may be possible that eelgrass beds might be able to buffer the effects of ocean acidification. So our hypothesis way back in 2010, we were seeing a long-term trend towards increasing pH in the bays and estuaries, thought that that might be due to local increases in eelgrass and algae. And so the hypothesis was that even though ocean waters were showing higher levels of PCO2 and lower DO and decreasing pH, once you got into that transition through the nearshore zone and up into the bays and estuaries, the situation became much more complicated. And we had to start to think about the net estuary ecosystem metabolism. So the dynamics inside the bays and estuaries are complicated. Since that time, over the next 12 years or so, these eelgrass beds at that particular location have all but disappeared, and the pH in the surrounding water has gone down. So even though it's been tragic to see the disappearance of that iconic eelgrass bed at South Slough, it's helping to prove this point that eelgrass beds may help buffer ocean acidification at a small local scale. What about in the nearshore zone? Respiration by giant kelp is also being identified as a possible way to buffer seawater and help alleviate ocean acidification. So work done down at Stanford University in Monterey Bay has looked at the dynamics and contribution of giant kelp, Macrocystis. And the findings there is that photosynthesis by the kelp, of course, produces oxygen and reduces carbon dioxide. And pH values were slightly higher in the surface water adjacent to the dense canopy of kelp up near the surface. The pH was lower, closer to the bottom, away from the kelp canopy. So that's a good lesson for us. But then here in Oregon, we don't have much Macrocystis, only one small bed down off Simpson Reef in Cape Arago. Most of our kelp is bull kelp, Nereocystis, and we're looking forward to learning how the photosynthesis and respiration by bull kelp may also play some kind of role in localized alteration of the biogeochemistry of seawater. So to just get close to wrapping this up, some fast facts. Ocean acidification causes changes in the carbonate chemistry of seawater. When carbonate saturation state, omega, decreases, seawater becomes corrosive to the shells of marine organisms. Second, the Pacific Northwest region is particularly vulnerable to ocean acidification due to intense coastal upwelling. Third, the biological impacts of ocean acidification, of course, are going to be variable from one species to the next. And fourth, that natural and anthropogenic factors combine to intensify ocean acidification in the Pacific Northwest waters. And then Pacific Northwest shellfish, of course, are particularly sensitive to reduced carbonate saturation within the current range of conditions that we see today. So I just want to leave you with this point that additional research and monitoring is needed to determine the impacts of ocean acidification and hypoxia to marine organisms, and also to understand the capabilities for adaptation and resilience. So the last point I want to make is that understanding the biological responses of our organisms is critical for us to be able to take responsible steps in coastal resource management. Beginning with the good understandings and detailed understandings that were generated at the Whiskey Creek Shellfish Hatchery, Oregon has taken a very interesting and fairly progressive pathway towards where we are now. So beginning back here on the left with recognizing those hatcheries failures through federal action and regional action here in the middle from 2009 through roughly 2016, and then formation here in Oregon in 2017 of the Oregon Ocean Acidification and Hypoxia Council, and then good work by that council as the forum for developing good progress here over the last 4 or 5 years. So that has really built upon these findings and knowledge bases that atmospheric CO2, of course, is a global problem, that ocean acidification along the West Coast is a regional intensification of a global problem that requires a coordinated regional approach, but that actions can be taken now to possibly mitigate, and that other actions can be taken now to support resilient ecosystems and allow ecosystems and communities and species to adapt, that we need to accelerate the development and integration of knowledge, have to conduct research to improve our understanding and to make better decisions, and that the cost of inaction will be very high. So just want to say hats off to our Ocean Acidification and Hypoxia Coordinating Council here in Oregon, a very broad group, tremendous expertise of folks who have been working on this for many years, and now come up with recently the second biennial report, and they're moving into some implementation stages. So 5 more pictures here, Oregon actions to address ocean acidification and hypoxia. One of those is to invest in Oregon's monitoring network to document oceanographic and biological conditions, as well as socioeconomic vulnerabilities related to ocean acidification and hypoxia. Second important action, to develop and integrate strategies to reduce the overall causes of excess carbon dioxide that contribute to ocean acidification and hypoxia. Third, to support activities and initiatives that promote adaptation and resilience to ocean acidification and hypoxia for Oregon's human communities, as well as our marine and estuarine ecosystems. Fourth, to focus on communicating ocean acidification and hypoxia, communicate the science, understanding of the impacts, and possible solutions to raise awareness and to support decision-making. And then finally, to mobilize our agencies to address ocean acidification and hypoxia as a priority emergent problem along our coast. So with that, I want to thank you for your attention and for this opportunity, and open it up for questions and pass back to Tara.
Thank you so much, Steve. That was really interesting. I really appreciated all the visuals that you shared. So for the audience, if you've got questions, please feel free to add them into the chat and the Q&A. Steve, I always like to kick off the Q&A with a question around inspiration. Like, what was your inspiration to kind of go deeper into studying shellfish and then later on ocean acidification as that became apparent?
Yeah, thanks for that question. Well, I'm very fortunate. I'm one of the few people in my network of colleagues who has been fortunate to grow up along the coastline and have some of my earliest memories as a child, walking along the coastline and shivering in the fog as my ankles and knees were being swept by the ocean. So I'm a coastal guy, and I grew up in the Central California area in that kind of situation. And then spent a lot of years of my childhood in Hawaii, where I was snorkeling and getting poked by sea urchins and catching sea cucumbers and all that kind of stuff. And then fortunate to just go right from there to focus in on becoming a professional marine biologist in college and throughout my degrees and my whole professional career. So it's been a one-track mind for me. So I don't really know where the inspiration came from. The shellfish side, I'm trained as a invertebrate zoologist, and as you can tell, I really appreciate the biology and ecology of marine invertebrate larvae. I'm really fascinated with the early life histories of marine invertebrates. But I was fortunate to take the leadership position at Oregon Department of Fish and Wildlife to run the shellfish program about a decade ago. And it's allowed me to focus on some very practical problems to ensure that we have sustainability of shellfish here for both recreational and commercial use, but to focus on fisheries management, on stock assessment, population biology, the early life histories and later life histories, longevity. It really just allowed me to focus in on these organisms rather than on a place. I enjoyed all the years that I worked at South Slough as an estuary ecosystem ecologist, and there it was more place focused, of which shellfish were a part of it. But now, these final parts of my career are really focusing on the marine invertebrates themselves and how they make a living and can survive in these stressful times.
Very cool. It's always nice to hear how it evolves into the career that you're currently doing. So our first question, this person, they've been noticing a lot of sea foam on the shore, especially after a storm. Can you speak to the amount of sea foam in relation to an acidified ocean?
Well, gosh, that's a great question. I don't know. So we typically think of sea foam as being formed by the mixture of exudates of diatoms and dinoflagellates, mostly diatoms. And, so these are microscopic algae that are in the ocean waters, and they're sloughing off materials as they live and break down. So there's a lot of kind of cellulose materials there in the water as well. And then mix that with winds and concentration, in particular coves like we see down at Devil's Churn, lots of foam right there. It really gets concentrated and thick. Now, during acidified situations like we're seeing now, as I mentioned, photosynthesis is not really affected, in fact, may actually be enhanced. So we could be seeing that situation where our diatom blooms are thicker and more robust. I don't know offhand, I'm not a phytoplankton person, but I don't know if those exudates of these cellulose materials are also enhanced by acidified ocean waters. But that's a good observation, a good question. We should ask our biological oceanographers to see if they can help with that. Yeah.
And then what about sea stars? Does ocean acidification have effect on the sea stars?
Yeah, so we're just beginning to really understand that. And so, we've seen this really tragic situation over the past 5 to 8 years on sea star wasting syndrome, killing off literally millions of sea stars, and some recovery of the ochre stars along the coastline, but really no recovery of our larger, very predatory multi-rayed, soft-bodied sea stars, like the sunflower star or the Solaster sea stars, cross aster, all of those sea stars. And there's some suspicion that ocean acidification may play a role. Typically, that was initially thought to be a virus, but and a parvo-type virus that contributed to the wasting of sea stars. But now more recent research is also looking at possible hypoxia, localized hypoxia at the level of the dermis, the skin that surrounds a sea star. And there may be some kind of role associated with acidified waters and localized hypoxia at the level of the body as well. Very detailed work trying to sort that out. Too early to just point the finger at ocean acidification, but definitely linked up with the effects of sea star wasting syndrome and marine heat wave. So a little curious that the observations of sea star wasting in Oregon lagged behind Washington and California. And we also recognize that Oregon also can have colder than normal, colder than regional seawater due to upwelling. So it's definitely tied to this in some kind of way. That's a great question. Yeah.
What actions do you think would be important for legislators to take to reduce CO2 emissions?
Yeah, so it's kind of a cliche, but this is really a global problem that has local impacts, global impacts and local impacts. So as Oregonians, we need to do our part to cut back on our CO2 emissions and decrease our global footprint, additions to greenhouse gas emissions. And Oregon is making good progress along those lines in terms of identifying what steps can be taken and trying to put together some progressive programs to help us along those ways. So we need to do our local part, and we're seeing changes to more fuel efficiency and solar power and looking at wave energy and alternates to greenhouse gas emissions. So all of those kind of contributions will help at the global level, but we need to just think that it does make a contribution at our shoreline. But of course, these are dissociated. The acidified ocean waters that upwell along the Oregon coast, it's likely that they've been underwater for decades. So we're seeing now in the conditions here as they upwell along our coast, a reflection of atmospheric CO2 10, 12, 13, 15 years ago in another part of the world, because we do have these belts of water, deep water moving underneath. So even if we stopped emitting greenhouse gas today, we're going to see the effects of these elevated CO2 levels in the atmosphere coming to the surface in acidified ocean waters for a long period of time.
We have many thanks rolling in, by the way. Just want to share that. And Rachel here shares that in her study is west of Ireland waters, and they don't have upwelling and hypoxia issues, but expect OA to become an issue for them in the longer term. Their research indicates that watershed geology and geology underlying the bays and estuaries that can be limestone, sandstone, or volcanic affects the pH and aragonite saturation of waters locally. Is there much geological variation along the Oregon coast? And is there any work on its possible impact on the OA status of local waters? Or would such effects be swamped by upwelling impacts?
That's a great question. I believe that, so thanks for pointing that out, that the local biogeochemistry of the underlying substratum does play a role, and unclear how important that role is along the Oregon coast. We do have quite a bit of geologic variability along our coast. Just right here in the Cape Perpetua area, a mix of basalt and old lava flows and sandstone and such, and river rock that has been deposited. So there's quite a bit of differences in the composition of the underlying bedrock. I don't know how much that influences the overlying water in the surf zone for us here, but we here in Oregon, we think of ourselves really along the central and southern Oregon coast as ground zero because of the driving importance of upwelling, delivering just massive amounts of acidified water. So I think that comparisons between upwelling centers like we have and other locations around the globe where we also see the effects of acidified ocean waters without upwelling will really give us some sort of feel. It's hard to do an experiment like that, but by comparing one geographic location to another, we can begin to dissect out the importance of underlying substratum versus upwelling and delivery of massive amounts of volumes of acidified water.
I always find it interesting how geology can make such an impact on things. Can you speak about the value of connectivity and resilience, and what can we do in regards to water quality?
Okay, so when I hear the word connectivity, I think, of course, everything is linked to each other. I jump to, of course, being a larval ecologist, to the connection between breeding and production of embryos and larvae, and then the drift of those biomaterials out in the plankton anywhere from 30 days, 60 days, 120 days to another location. That's very important when we think about the connectivity of biomaterials from one protected area to another, or an area that is experiencing heavy fish out and extraction of fish or invertebrates to another area, a source and a sink. So that connectivity is very important on biological populations. It's also important when you think about the resilience of those populations that are a sink for connectivity, that receive planktonic larvae coming from other locations, can withstand pretty high levels of extraction because they're continually getting input of young juveniles into the system, and they're not reliant on the local production. So connectivity and resilience are super important just from biomaterials. And you think about that from connectivity of other pollutants, legacy contaminants, oil and grease and heavy metals, or more contemporary understanding of the role of microplastics produced in one part of the world and drifting to another. The same source sink kind of dynamics will also come into play there. It really, there's a whole science in terms of trying to map these connectivity pathways. And the larval fish people are doing a great job at that, trying to track where the source populations are and where those offspring are going. I used to, when I talked about this decades ago, I used to hold up a clamshell and say, where are my parents? Or where did my kids go? That kind of thing. We could do the same thing with a crab. Just hold that up today and say, where are my parents? Where did my kids go? And a lot of that is done by the combination of physical, biological oceanography, tracking the currents, their strength and the directions, tracking those, as well as computer modeling. Great centers for that here in Oregon. I'm not an expert in it, but I understand enough just to know that it's important.
Mm-hmm. And speaking of microplastics, does an increase in microplastic waste cause an increase in acidification?
Well, of course, microplastics, of course, are really complicated as well. So the composition of the plastics can be very variable, and the biofilms that are attracted to the surface of microplastics also develop their own microenvironment. I don't know how that actually works, but you could think that diatom scum and such that could develop on the surface of mesoplastics could be enhanced by acidified ocean waters. At the nanoplastic level, below microplastics, we're too small really to have diatoms attached to them, but they're really going to be characterized by the composition of the underlying plastic type. I'm not sure how that chemistry would work, but as we move from what I think of as big marine debris that can develop whole marine communities attached to it, down through smaller and smaller and smaller microplastics, down to nanoplastics, I think the impacts with shifts in the chemistry of ocean water will become more and more important.
Mm-hmm. Do you know what caused the disappearance of eelgrass at the South Slough?
No, so as part of the eelgrass mafia, we watch these sites, and it's very unfortunate. That site was established as one of the regional global seagrass monitoring stations, and have a long legacy of observations at that area. And it has undergone this rapid decline. Other locations in the Coos Estuary have not, though. So it's something particular about that location, and it could be related to a shift in the chemistry of water there, could be related to a shift in the overall bathymetry of the site, sediment loading, could be biotic issues associated with eelgrass disease. So we're not sure exactly what's gone on there, but it's a focus for some intense monitoring and study and research.
All right, and last question of the day. Are the same problems for shellfish being seen on the East Coast of the US?
Yeah, so ocean acidification is a global problem, and it's really just the, the play, the storyline for the play is being told by different players. And we talked a little bit about that in terms of the importance of the geomorphology of the oceanography along the coastline, but the marine invertebrates and fish are different there as well. We're definitely seeing the impacts of ocean acidification almost everywhere that we look along the coastline. It's just different players in that story.
Very cool. Well, again, Steve, I really appreciate you joining us this morning. Many folks have commented, and thank you for the presentation. And very informative and inspirational. So really appreciate that again. Thank you to everybody who joined us today, and hope to see you at a future presentation.
All right. Thanks so much, Shara, and thanks to everyone in the audience for those great questions and for listening to me tell the story. I really appreciate the chance to share that. And once again, it was an honor to be your speaker here today on Ocean Acidification Day of Action. So hopefully this will stick with you throughout the day and think about this problem. Thanks very much.
Have a great weekend, everyone.